From the Python project, we have a course called CRISPR Mutagenesis in Xenopus. We also have a course called the Discovery Lab, phage genomics, and the cell biology laboratory. So all of you here are going to be presenting your real research, which is really incredible. If you haven't gotten instructions on who you're evaluating, please see your instructor. You'll be presenting in one session, one poster session, and you'll be evaluating in the other poster session. So I think I probably speak for all of the instructors when I say that we are so proud of what you've all accomplished. This is a huge deal. The courses that you're taking this semester either just started or being offered for the first time at such a big scale. So there are 273 of you right now taking research courses in MCDB. I know when I started teaching these courses, I thought, there's no way, freshmen can't do research, but we'll try, right? And I sort of thought of myself as a tour guide, right? And we're giving you a tour on a tour bus of one of the coolest places, which is research. And I think that some of us expected you guys to kind of hang out on the bus and take pictures and post them on Facebook and be like, yeah, this is really cool. And on the other hand, we expected a small number of you to get off the bus and interact with people and speak the language and try to buy things at stores and be part of the culture. And I think that what you've proven is that all of you have done that. Today, you're communicating research to other researchers, which is so cool. So enjoy the day. We're going to start out with a few speakers. We have one talk from each of the courses that are represented here. And I'll turn it over to Christy Philman, who will introduce our first speaker. First, I should mention, though, that this wouldn't be possible without our many sponsors. So the Howard Hughes Medical Institute, for one, the Chancellor of the University has given support, for example, to the CRISPR mutagenesis class, the Department of Molecular, Cellular, and Developmental Biology, and ESI, the Biological Sciences Initiative. But you guys wouldn't be here without them. And there are also many private donors who have donated money to make the research possible. So I'll turn it over to Christy, and at the end of each talk, if you can just think of some questions, we'll take one or two questions for each of the presenters.
Suspected to be in the C1 cluster.
Hi everyone, I'm Chad. I'm Matthew. So, to begin, what you see on the right, that sort of alien-looking structure, that's a bacteriophage. These guys, they infect bacteria. They're viruses for a specific host bacteria. So, why is this important? So as probably many of you know, drug-resistant bacteria, specifically multi-drug-resistant tuberculosis, is sort of becoming more and more prevalent in the health scene. And as such, mycobacteriophages, or bacteriophages in general, are being looked at as a potential cure for drug-resistant infection. So the purpose of this class was to isolate and characterize a novel mycobacteriophage for the purpose of accelerating the study of bacteriophages worldwide. So in this class, specifically between Matthew and I, we isolated mini-me, and we suspected it to be in cluster C1. So the host that we used to isolate our bacteriophage was Mycobacterium schmegmatis, which has a funny name. This mycobacterium is actually a surrogate for Mycobacterium tuberculosis. So it allows us to study potential cures for tuberculosis without using it in the lab. And plus, schmegmatis is a great host. So, just to be general, a cluster is a way of organizing the vast amount of bacteriophages into different similarity groups, and those similarities are based on plaque morphology, electron microscopy, a restriction digest, and also genome similarity.
So there are many different phages. There are two different life cycles that phage can undergo, lytic and lysogenic. In the lytic cycle, the phage attaches to the host cells, and then it inserts its genetic information, taking over the cell's machinery, producing more phage parts, and eventually lysing the cell. And in the lysogenic cycle, the DNA, its genetic information is actually inserted into the host's genetic information, and then can go through the cell reproduction cycle. Then eventually, once conditions become unworthy for the phage, the phage can't sustain it anymore. It actually excises its genetic information out of the host's genome, chromosomes, and then starts producing phage parts and eventually lysing the cell. A way we can visualize these two different life cycles are plaques. Plaques are small clearings on the long bacteria. A lytic phage develops clear plaques, seen here. All of the host cells are killed, and a lysogenic plaque would be turbid, as all the host cells are killed, but only some. And these are plaques from mini-me, and as you can see, they are very clear, meaning it is a lytic phage.
So there are also two different structures of bacteriophages. There's myoviridae and cycloviridae. So myoviridae have large heads with short contractile tails, and cycloviridae have small heads with long tails that are non-contractile. So mini-me is actually a myoviridae phage. So that's mini-me on the right. It had a 100 nanometer in diameter head with a short 85 nanometer tail, and this compared favorably with Cali, which is a known cluster C1 phage.
Another way we visualized what this phage mini-me was through a restriction digestion analysis. So after we had isolated phage mini-me, we removed its DNA and cut it with 5 restriction enzymes. And from that, there are 2 important things we can see on the restriction digestion. It's the cuts from BAMH1 and CLAW1. The orange yellowish arrow points to BAMH1, and specifically what we noticed that was interesting was how very few cuts there were, and also how many cuts were on CLAW1, and that is a characteristic of C cluster phage, specifically the C1 phage. We compare that to C cluster phage Cali.
So after with data from the plaque morphology, the electron micrograph, and restriction digest, we built the hypothesis that mini-me was a C1 cluster phage. So it would be cluster C and then subcluster C1. And as such, we decided to run polymerase chain reaction, PCR, with C1 specific primers. So these primers will only anneal to C1 phages because there's a specific sequence in each of those. Sorry, excuse me. So if you look, the one the arrow is pointing to is mini-me DNA after it was run with C1 specific primers. And then, and then on the left side of that, we have the positive control, which was actually a C1 primer with a C1 phage. So we sort of had the double guarantee that it was a C1 phage.
So with all the information we collected from our plaque morphology, being a lytic phage, developing clear plaques, the electron micrograph showing visually it is similar to a C cluster phage, the restriction digestion analysis showing it has key features that C cluster phage do, and also our test with C1 primers, we do believe it is a C1 cluster phage. That's all the information we have.
So we are happy to say that mini-me was selected by our class to go to the Howard Hughes Medical Institute for genome sequencing. And with this information, we would like to confirm that mini-me is actually a C1 cluster phage, because ultimately the only way to absolutely confirm cluster is genome sequencing. So for a phage to be in the same cluster, it would have to have 50% gene similarity with others in that cluster. And for it to be in the same subcluster, it would have to have 75% gene similarity with others in that subcluster. So, and then another thing we would like to answer would be why the plaques are so small, because in other bacteriophages there's very large plaques. And we believe, our hypothesis is that Mini-Me has actually a very large genome, which means it takes longer to replicate, and thus the plaques are smaller. And we'd also like to know that why the genetic reason is that only C cluster phages are myeloviridae. That's how we knew that Mini-Me was likely a C cluster phage because of its shape. So there are also many applications to bacteriophage research, as we stated at the beginning. The first of which is clinical and therapeutic. So we found research based on clinical and therapeutic, where bacteriophages were used as reporters for multi-drug resistant tuberculosis infection in high HIV burden populations. So these bacteriophages would bioluminesce once they encountered a tuberculosis bacteria. And that would allow us for greater certainty in diagnosing tuberculosis, because up until, or currently, it is a semi-inaccurate process.
So another thing we found out is they can also be used not just in humans, but in animals too. So phage have been known to kill many diseases, and in Indiana, 14 different phage were isolated, and a cocktail was produced from that, given to swine, and there was a 99% reduction in salmonella within its intestinal system. So we see that you can see that there is a potential for reducing infections and diseases in humans, but also in animals. This can affect food safety and just a multitude of other things.
So bacteriophages represent a wealth of information. We have some great names. All of these bacteriophages were isolated in the Dirty DNA phage genomics lab. Plus there's many more. This is just a few. So we believe that bacteriophages have broad applications worldwide, and they can affect many different sectors. And the research is just beginning, and that's what we're here to do. Bacteriophages represent a vastly unstudied hub of genetic information, and we're excited that we were able to research them.
Lastly, we would like to acknowledge Dr. Tillman, Dr. Dial, Josh Keys, Saravia Karmanchi, the University of Colorado Boulder, and the Howard Hughes Medical Institute for the support of funding.
Do you have any questions?
So you said there was an application on food safety. How was the delivery method?
It was just, they injected basically approximately 14 different phages in the solution into the swine, which then worked through the swine body, just effectively killing all the salmonella bacteria inside the pig. So that's go ahead.
Did you guys ever investigate whether C1 clusters had small plaques and large genomes, or is it possible that maybe might be a new subcluster?
It's definitely possible that Mini-Me could be a new cluster, but how we developed our hypothesis that it was C1 was by comparing it to the database of other C1 bacteriophages on the database. So we're pretty confident that it is, but there is definitely a possibility, and that would be answered with genome sequencing.
All right, let's thank our speakers. So our next presentation is from Rachel Anderson, who's from the Drug Discovery Lab, and she'll be talking about structure-based prediction of novel antibiotic targets.
Okay, all right, awesome. Hi, so I'm Rachel Anderson, and I am a student in the Discovery Lab, and I'm going to be talking today about our work with salmonella. So let's get started. What do you think of when you hear salmonella? Maybe you think of swine intestines or chickens and their eggs. That's not what I'm talking about. I'm talking about a much worse form of salmonella, Salmonella typhi, which causes typhoid fever. Typhoid fever is a pretty nasty illness that affects 21 million people a year and kills about 220,000. If you were to come down with salmonella or typhoid fever today, you'd be bedridden for up to a month with severe fever, delirium, gastrointestinal issues, and there's about a 20% chance that even with treatment you would die. Unless, of course, you took antibiotics. Antibiotics are a great treatment, but they may not always be an option. Right now, 70% of typhoid cases are resistant to one form of antibiotic, and 10% are resistant to 3 or more forms of antibiotic. So what do I mean when I say resistant, and why does that matter? There's a ton of bacteria spread all around us, and when you take an antibiotic because you're sick, it kills most of the bacteria except for those that have naturally occurring immunity. If you don't clear your infection, then those bacteria can then replicate, they can spread their immunity to other bacteria, even other species, which means that the next person who gets sick and tries to take the drug, it no longer works. That should be pretty alarming when you consider how fast antibiotic resistance is increasing. Largely due to the amount of antibiotics that we use in agriculture. More than half of antibiotics used in the United States are on agriculture, like agricultural aspects like cattle, chickens, things like that. This is a big problem, especially when antibiotics resistance is predicted to cause more deaths than cancer by the year 2050. So we need some way of fixing this before it gets out of hand. So let's find some new antibiotics. How do we find them? Well, you can randomly stumble into them, like Alexander Fleming and penicillin, or you can do large-scale screens of tens of thousands of compounds and try to find one that actually kills a bacteria, or you can do a targeted approach, where you pick something on the cells, like a cell wall or a protein synthesis, and you say, I want to stop this from happening. We tried a slightly hybrid approach where we screened the library under unique conditions. It was a pretty interesting library. So let's talk about that. So before we do that, though, we need to talk about salmonella itself. So inside of our immune system, we have these cells called macrophages, which literally engulf and then digest intruders in our body. So salmonella kind of outsmart this process. They do get engulfed, but then once they're inside the macrophage, they prevent digestion. And they just sit inside the macrophage, merrily reproducing, outwitting our own body's defenses. So these are really harsh conditions for salmonella to be in. It's a very low pH, it's acidic, there's not a lot of access to nutrients. So we wanted to study salmonella under really similar conditions. So we used something called M9 minimal media, which is basically just starving the salmonella. They're really feeling threatened. It's a great way to mimic the conditions in the body. So then we also got a 1,600 compound library from the National Cancer Institute, which is a group of really structurally unique compounds. And then we took those compounds and we took these 96 well plates, and to every single one of those wells we would add salmonella, and then one of a single compound from the library, or dimethyl sulfoxide, the solvent for the compounds, which is our negative control. We don't expect that to do anything. Or ampicillin, our positive control, which we expect to kill all the bacteria. Then we took the plates and we incubated them for 24 hours at body temperature, again mimicking natural growth conditions. And then we quantified the growth of salmonella by spectrophotometry, which is basically just shining a light through the plate and seeing how much is absorbed. So what did we find? Well, we got a lot of data. And then we crunched the data. And I love this graph just because it shows how much data we got. So every single one of these points is an individual compound trial. And you can see that most of them fall within 2 standard deviations of the mean. So they either don't do much of anything or they have a small effect. But there's a good number of compounds that fall outside of those range. And those are either really inhibit or really promote the growth of salmonella. And those are the ones we wanted to study further because those actually have great applications. So we found 22 compounds in total that inhibited growth of Salmonella as effectively as our positive control ampicillin here in green. Those are the compounds we want to study. Those could potentially one day be the next antibiotic on the market. We did also find 6 compounds that promoted growth, which it's not as useful to us because we don't want salmonella to grow, but it's something we need to know about because if, say, those were in a household cleaner, you don't want to be spraying that everywhere, increasing the growth of salmonella. So then from here, we split off into our own individual projects, and I'll talk about mine, which was dealing more with structure. So, excuse me. So when I looked at all of our 22 hits, I noticed that there were a lot of structural similarities. And I wanted to know if I could make predictions about new antibiotics based on their similar structure to our compounds. So what I did was I grouped our hits by structural similarity, and then I looked at existing classes of antibiotics that were already on the market. And I tried to make some educated guesses about which functional groups were chemically responsible for the activity that we saw. So I prepared a small example here. If you're not great with organic chemistry, you can just look at the regions I've highlighted in red. So you can see that compound 1 is a hit from our screen that prevented the growth of Salmonella. You have 2 aromatic rings with a diamide linker in the middle. Compound 2 is triclocarban. That's an antibiotic found in hand soap. Again, you have the same general structure. There's different groups in the periphery, but it's the same overall structure. Then compound 3 is a compound I hand-screened from a different library that I was hoping would prove to be effective as an antibiotic. It's not exactly the same. I've swapped out they swapped out the oxygen for nitrogen, but it's the same general idea. So then I picked 8 of these compounds through my screen, and I did a dosing series to see if they would be effective as antibiotics. And unfortunately, they weren't. They were all duds. They were all very mediocre at actually preventing growth. So why is that? What went wrong here? To understand that, I feel like we need to step back and look at what happens in the biological system when you take a compound. So instead of antibiotics, which are kind of hazy, let's talk about pain relief. You've all probably heard of morphine, which is a pretty strong pain reliever. Fentanyl is a much stronger pain reliever. It's about, I think it's about 1,000 times stronger. And I put the structure up here. And then carfentanil is a derivative of fentanyl, which is 100 times stronger than fentanyl itself. And the only thing that's changed is that little tiny group where they've added an ester highlighted in red. So a tiny structural change can produce a massive effect. So that's what I think happened here. With my compounds, a tiny structural change had reduced the efficacy of my antibiotics by such a large degree that they no longer inhibited growth effectively. So, where do we go from here? Well, all our data will be forwarded to the Detweiler lab, and they'll do cool things like cell assays, maybe test some compounds with mice if they're really promising, and stuff like that. But of course, the Discovery Lab itself doesn't end here. There will be more students who come through, and they can look at our research and build off of it, and maybe find something that we missed, or who knows what they'll do. So, to recap, typhoid fever, caused by Salmonella typhi. Pretty nasty illness, affects about 21 million people a year and kills 220,000. It's antibiotic resistance in Salmonella typhi is a problem, and it's getting worse. We know that about 70% are resistant to one drug, and 10% are resistant to 3 drugs or more. So we need new antibiotics. So to help work with this, we screened nearly 1,600 compounds, and we found 22 that inhibited growth. And I also found that predicting structure activity is hard. It's a really useful tool in medicinal chemistry, but it's actually surprisingly hard. So I would like to say thank you to our sponsors, the Howard Hughes Medical Institute, the MCDB department, and the Biological Science Initiative, because without them, the Discovery Lab would not have been possible. So, thank you. You know what, I don't don't entirely know that. I do know it's a very acidic environment. I would expect that some of them do just get chewed up because it's so acidic, but I'm guessing that they've just evolved over time to be really resistant to acid and potentially just repair damage. All right, thank you.
Our next speakers are Kimberly Lugo, Jessica Miller, and Philip Rubin. They are from the cell biology course, and they'll be talking about mutated oral growth and enhanced phagocytic rate in NP1 tetrahymena Drosophila.
Hi, I'm Phil.
I'm Jessica. And I'm Kimberly Lugo, and we'll be presenting to you our research on NP1 mutant strain tetrahymena. So, this semester, the cellular biology lab course had several groups of students who investigated tetrahymena in different strains or different deformities that they wanted to investigate using different microscope techniques that we learned throughout the semester. A little bit of background about the tetrahymena is that they're a single-celled organism who inhabits freshwater ecosystems like streams, lakes, and ponds, and they're about 40 to 50 micrometers in length. What makes this organism so ideal for study and why it was our center of research in the cellular biology lab is that they're relatively easy to maintain and culture from week to week. They're inexpensive, and their complex processes that they carry out are conserved over a wide range of eukaryotic cell types, which makes our research applicable to a lot of different fields. Some relevant structures that we were investigated in our experiment were basal bodies. Now, basal bodies are these little barrel-shaped proteins that associate on the surface of the cell, and they form these long linear structures, as you can see from the anterior end to the posterior end in green, and they make these long linear structures that provide support to the outside cytoskeletal structure of the tetrahymena. Not only do they provide structural support to the tetrahymena, they provide a surface to where cilia can anneal. And cilia are hair-like structures that, when they anneal to these basal bodies, they're allowed to move in the water, which provides a current in the tetrahymena and allows them to move around in their surroundings. As well as the cilia help carry out many different sensory functions as well. Now, the oral groove is that red puckered structure that you see toward the anterior end, and the oral groove is a really concentrated structure of basal bodies and cilia. And when these coordinated cilia start beating, it allows food particles to be brought near the oral groove. And when the food particles are brought near the oral groove, a food vacuole can then surround the food particle and bring the food vacuole into the cell, where it can further degrade this particle for different metabolic processes in a process called phagocytosis, which was one of the main points of our experiment.
Okay, so when our group started looking into the functionality of tetrahymena as a model organism, our group was particularly interested in the NP1 mutant strain. The NP1 strain was characterized to show maldevelopment, temperature-sensitive maldevelopment in the oral groove development, and that would occur at 37 degrees Celsius. And unfortunately, this was all we were able to find about the mutation. We were unable to find a more molecular background, and that led us to being curious about how this mutation was induced, whether it was induced rapidly, directly at 37 degrees Celsius, or whether you would see the mutation manifest as the temperature would increase. We decided to use a few different approaches to direct our research when we're looking at this question. First off, we decided to measure the rates of phagocytosis in the NP1 mutant strain and the wild type at different temperatures. We did this by adding India ink to cell cultures and fixing aliquots at set time intervals in order to visualize the food vacuoles that had formed. If you look at the bottom right picture, you can see the India ink food vacuoles that would form over time. Another approach that we used, we learned how to do the immunocytochemistry procedure, in which you fluorescently label specific proteins using antibodies. This would allow us to visualize the particular proteins in the cell that we were interested in. We decided to fluorescently label centrin, which is a protein involved in the basal body, which was discussed earlier. As you can see in the upper right, this was actually an immunocytochemistry picture that we had taken. And this would allow us to visualize the cytoskeletal phenotype of the cell. So we ended up hypothesizing that the NP1 mutant strain would demonstrate fewer food vacuoles forming, especially at temperatures above 37 degrees, when we predicted that the mutation would be induced. We also predicted that the fluorescent imaging of the cytoskeleton would show some aberrant structure in the organization of basal bodies, which we could then visualize.
So the first part of our experimentation was phagocytosis, where we took samples of wild-type and NP1 mutant tetrahymena and incubated them using a few different methods, an incubator and heat clock. And after they had adjusted to the temperature, we added India ink and then took samples at 3, 5, 10, and 15-minute intervals after incubation in India ink. And we fixed those samples and counted the number of food vacuoles present across 25 different cells to obtain the average number of food vacuoles per cell. And our graphs here display the 10-minute time point incubation. So on the left, we have incubation in an incubator, and you can see at both temperature points, the NP1 mutant shows more food vacuoles. And this theme holds true after our incubation in the heat block. At all of those various temperature points, the NP1 mutant has more food vacuoles per cell than the wild type. And this was initially very counterintuitive to our understanding of the mutation. We knew that the malformation of the oral groove occurred at 37 degrees Celsius, but we were surprised to see that a mutant would have increased rates of phagocytosis at all temperature points leading up to that, as opposed to the wild type. But when we took a look at our immunocytochemistry, this kind of supported our findings in phagocytosis. So here, as mentioned before, we performed immunocytochemistry and we labeled centrin protein in basal bodies, which shows up as a red fluorescence. So here you'll see the basal bodies highlighted really nicely by that fluorescent labeling. On the left is the NP1 mutant incubated at room temp, so no temperature increase. We're not expecting to see the knockout at all. But we have a very distinct elongated shape with a kind of concavity near the oral groove that you're not seeing in the wild type, which is there on the right. Also incubated at room temp, but it has a far more rounded shape and it doesn't really have that concavity near the oral groove. So analyzing both of these images shows us that there's a very distinct phenotypic difference in the two cells without any temperature increase. There's the cell shapes show that there is a difference in phagocytosis, and this supports what we saw in our phagocytosis data. And then our next slide shows a little more zoomed in image of the oral groove. So once again, NP1 incubated at room temperature on the left. We see a really distinct concavity, and then wild type on the right is a far more convex shape. So once again, we do see a very distinct phenotypic difference in overall cell shape, especially pertaining to the oral groove. And we can infer that this is what we saw in our phagocytosis data that wasn't as we expected. Something about this phenotypic difference may make it easier for the NP1 mutant to perform phagocytosis and therefore have more feed vacuoles.
So just to reiterate, our objective was to look into how temperature would affect the induction of this NP1 mutation and how the cytoskeleton would phenotypically look different from the wild type.
Yeah, and our results didn't really tell us that much about malformation of the oral groove at 37 degrees Celsius, but we did still get some really interesting results at our different temperature points. Across the board, we saw increased phagocytosis in the NP1 mutant as opposed to the wild type. And this was supported when we looked at our immunocytochemistry data and saw that there's a very distinct phenotypic difference in the shape of the two cells. The NP1 is more elongated and has a more convex oral groove, whereas concave oral groove, whereas the wild type is a lot more rounded and doesn't have that distinct difference in skull groove.
Regarding future directions for this experiment, we would love future students of the cellular biology lab to continue to look into the NP1 mutant because there's a lot. We would like to repeat the phagocytosis procedure that we outlined again, just to further solidify our findings and just have more data to bounce back on. And also repeat the immunocytochemistry experiment because we weren't able to obtain an NP1 fluorescently labeled image at 37 degrees. And we would really like to see at 37 and varying time points around there how this more oral groove malformation is induced, and if it continues over certain time points or if it changes phenotype at all. That would be something we would really like to investigate. And we would like to keep the tetrahymena in a heated incubator at several different time points for several weeks to see if that malformation of the oral groove is carried on through successive generations. We would also like to assess the mutants as well as the wild type selectivity toward different environmental particles, because even though the NP1 mutant had increased rates of phagocytosis with regards to India ink, it would be interesting to see if they have certain selectivity in regards to size or pH or different environmental stimuli. We would just like to acknowledge Jerry Hans for providing us with the NP1 mutant strain, Alex Stemwolf for providing the anti-centrin antibodies so we could visualize ourselves, as well as Joy Power for giving us all the necessary tools we needed throughout the semester, and Jesse and William for our TAs for making the labs a fun learning environment this semester. Thanks.
And the NP1 was able to survive that in such a firm process.
Could you sort of increase rates of phagocytosis in the NP1 mutant at 8 degrees Celsius? Is it possible that the phenotype of the NP1 mutant is inherently more suited to phagocytosis compared to wild type? Is it possible that there's something else besides the oral groove that cause it to increase rate?
Yeah, we kind of want to look into that more, but I mean, what we could really draw from what we did in our experimentation is that we did see a difference in shape of the cell, so we just assumed that that's what helped with the increased rate of phagocytosis.
Great, thank you. The next speaker is Julia Moon. She is from the CRISPR mutagenesis in Xenopus lab, and she'll be talking about mutagenesis of TPR-G1L in Xenopus laevis.
My name is Julia Moon, and I'm going to be talking about the mutagenesis of Xenopus laevis in regards to the gene TPR-G1. And Xenopus laevis is also known as the African clod frog, if you ever get confused midway through. So a big question in developmental biology is how exactly do we get from a single cell embryo all the way to this complex multicellular organism that we are today? And it involves a lot of coordination between these genetic molecules telling each other what to do. And when one of those molecules is missing, or there's too much of it or too little of it, you end up with genetic disorders such as Down syndrome, sickle cell anemia, color blindness, ectrodactyly, or Duchenne's muscular dystrophy, to name a few. So figuring out what genes do help us figure out these types of diseases. And a way for biologists to figure out how genes work is to find a gene, mutate it so it's not functional. Then they see what kind of like weird stuff happens, and then through that they figure out what that gene does. And understanding what exactly genes do help us provide treatments. So the first objective was to find a gene with an unknown function in frogs, successfully mutate it, see what happens, and hopefully figure out what it does. So the process was 2 steps. The first being the mutagenesis using CRISPR-Cas9. And CRISPR-Cas9 is basically a genome editing tool that we would inject into embryos, and that would inhibit proper gene function of the tryptophan gene. And the second being in situ hybridization, so that just shows me where that gene was expressed inside my embryo, so both of them work together to figure out what it does. So CRISPR-Cas9 is this new cool kid on the block. It's both faster and more efficient than any other genome editing tool out on the market today, such as your talons or your zinc finger nucleases. It has these 2 main components to it, the first of which being the guide RNA, which is a small 20 nucleotide sequence that scans through the organism's genomic DNA, finds a complementary pairing, and then Cas9 is an endonuclease that it's bound to. So it just cuts the DNA where that is, and then the cell freaks out, and it calls upon its endogenous cell repair mechanisms, one of which being non-homologous end joining. That creates a lot of insertions, deletions, or just frameshift mutations, all of which inhibit the proper function of the gene. So Xanthus flavus was chosen as the model organism due to its low associated cost per embryo. It's rather large embryo size. It's 1 to 1.3 millimeters in diameter, so it's really easy to inject it. And for comparison, it's around 10 times larger than the human egg. They also developed externally, which also allows for easy manipulation, so you don't have to put them back in the uterus. And they also grow really, really quickly. You can get from a single cell oocyte all the way to a tadpole within 3 days, and all the vital organs and structures have already been developed, so it's pretty easy and fast to figure out what the mutagenesis experiment leads to. And they also have a super high genome structural similarity to humans, so any gene function that we find in frogs can provide insight into what it does in humans. So more about the gene that I chose, it's also known as tuber protein P63 regulated 1 life, or TPRG1. I've scoured through the internet and I found 3 main papers that discuss it. The first of which describes TPRG1 as a gene that codes for this presynaptic protein. It lives in between that synapse right there, so it is a part of your brain function. It is also a potential marker for familial idiopathic scoliosis, which is a skeletal disease that leads in a lateral S curve in someone's spine. And it's also a marker for microRNA-21, which is highly expressed in many types of tumor cells. So the function of TPRG1 in Xanthus flavus is actually unknown. I pulled this from Zenbase, which covers basically anything about frogs. And as you can see, all we know is its acronym and its name, so it's great. So the first part of my mutagenesis experiment was to identify a target sequence that my CRISPR-Cas9 system would target. I chose the first exon because once cut, it would alter anything downstream of it, which would thus inhibit its full function. Along with that, the DR274 plasmid was the plasmid that I chose to own it in due to its T7 RNA polymerase promoter site. So once the target sequence was ligated into the linearized vector, I did a bacterial transformation so I could propagate more DNA to work with. And then I picked a colony that tested positive for my construct, isolated that plasmid out, and then did a restriction digest on it, which allowed me to have this specific type of construct that started with a T7 RNA polymerase promoter sequence, followed by my target sequence, and then a scaffolding sequence that was essential for the Cas9 to bind to it. So the T7 RNA polymerase promoter sequence just allowed for the T7 RNA polymerase to bind to it, and then transcribe and poop out tons of sgRNA for me to work with. So once I had my sgRNA, I made a sgRNA and Cas9 complex, then used some needles, injected them into embryos and waited to see if any mutants occurred. Unfortunately, my results did not end in a head-butt tail thing. Any mutant phenotypes that arose, I took a note of it, genotype to make sure that it was in fact that specific gene that was mutated and induced that phenotype. Or if there was no phenotype, I'd also genotype that to make sure, like, all right, I did what I did and it still did nothing. The second part I was mentioning was the in situ hybridization. So that requires me to make this construct that from the cDNA sequence, so that's basically all the exons inside the gene, found the complementary sequence for the mRNA endogenous to the organism, hybridize that to a fixed embryo, and those purple blobs would indicate exactly where it's expressed. So that would provide me with the visualization, patternization of the gene expression. So, in conclusion, my TPR gene knocked out mutant phenotype, along with my expression pattern, would provide a function for PPR gene synthesis. I'm still working on it. I tried 2 mutagenesis experiments, the first of which resulted in a couple embryos with axis deficiency, and the second of which just died. I fully amplified the cDNA length, so in situ hybridization should be coming along soon enough. If you have any more questions, I'd love to